This is my eReport for Unit 1 for Cellular and Molecular Biology. The topic outlines we'll be covering today are going to be in the exact order that they appear on the screen. So firstly, we'll talk about chromatin compaction, what it means to be chromatin, how do we fit all this DNA into a nucleus, what are the mechanisms that we can use to achieve localized DNA accessibility and inaccessibility. Then we'll talk about DNA replication. We'll talk about every step from the origin of replication to the bubble, to helicase, to the fork, to the synthesis itself, what DNA polymerase is, what it means to be leading versus lagging, what a primer is, Okazaki, everything involved with replication. Then we'll jump into DNA mismatch repair and dsDNA breaks and repairs. DNA mismatch repair kind of has a nice lead-in from DNA replication because of something that DNA polymerase can do aside from synthesized DNA. I kind of refer to this as the safety net mismatch repair, or safety net repair mechanism, I should say, and we'll see why later. We'll talk about dsDNA breaks, we'll discuss why they're lethal, and we'll talk about the two mechanisms of repair that we can use, and by we I mean the cell, that the cell can use to fix these breaks. Then we'll jump into the PIC, or the pre-initiator complex. We'll talk about what transcription factors come into play, how they recruit RNA polymerase II for transcription. We'll even discuss the order that they come in and why they come in. Then we'll move on to post-transcriptional mRNA processing, and this is just a quality control type of deal for mRNA in the nucleus. Before it can leave the nucleus, it has to be given these seals of approval, or verification stickers, I should say, tagging it saying that this mRNA is mature and genuine and is ready to leave the nucleus. We'll talk about what that means in a little bit. Then we'll hit up on translation, and when I say translation, I mean all of it. We'll discuss the amino acids, we'll discuss what start and stop codons are, we'll discuss the reading frames, we'll discuss tRNA, the large and small ribosomal subunits, what role ribosomes play in translation, the A site, E site, P site, polypeptide chains, and release factors. We'll just cover all of it as much as we can. Then we'll hop into combinatorial control. In the mediator complex, we'll talk about what combinatorial control is meant to do for the cell, how it's supposed to help with RNA polymerase II. We'll discuss what mediator complex is meant to do, where it sits, what stage are we in the cell, how they tie together, and what they're supposed to do. What is their purpose? And then we'll wrap it up with RNAi, or RNA interference. We'll discuss what that mechanism is made for. We'll talk about double-stranded RNA, how it's made. We'll talk about dicer, RISC, siRNA, amplification, RDRP, and everything that goes underneath that. So before we jump directly into topic number 1, we just want to know what we're dealing with here. So the human genome is 6 feet long. That's a lot of DNA, and we've got to find a way to fit this into the nucleus, which is incredibly tiny. This is a real-life tangible example. It's like trying to fit 24 miles worth of thin thread into a hollow tennis ball. That is just mind-boggling. The fact that we can even conceptualize fitting 24 miles worth of thin thread into a hollow tennis ball, and compare that to 6 feet of DNA into a tiny, tiny nucleus that cannot be seen with the naked eye. That is just insane. I love that our cells are able to do this. They don't know how because cells don't know anything. But the fact that they're able to makes us want to learn more. Oh, my stylus. Oh gosh. So here we go. Topic 1. So what is the general idea that we're getting into here? So we start off with the beginning of the condensing stage. And this is where the histone wheel, also known as the nucleosome, which is made up of 4 separate histone proteins that double themselves and latch onto DNA. This DNA will then wrap around the nucleosome, and each nucleosome will stack on top of one another, and DNA will still continuously be wrapping around them. This is going to form the 30 nanometer fiber, and then from this, DNA can become either accessible or inaccessible with the help of 2 different complexes. That's a lot of words, so we're just going to jump into a little bit more detail, but a lot more slow. So once these histone wheels have the DNA wrapped, it's going to become a certain uniform shape, and that shape will take a certain diameter, and that diameter is 30 nanometers. This is where we get the 30 nanometer fiber, and the formation of this comes from the H1 protein. This protein is what allows DNA to coil appropriately. Imagine you have a garden hose and you want to twist it a certain way so that it'll bend the way you want it to. The H1 protein is what allows you to do that. It will allow the DNA or allow your garden hose to turn the way you want it to. Another example I like to use to kind of illustrate the 30 nanometer fiber, though, is this spring. This spring has a consistent diameter throughout, and the shape that it takes almost reminds me of the DNA, wrapping around the nucleosomes continuously. Now imagine if we had a metal rod that kind of shot through the middle here. That metal rod that the spring is wrapping itself around would be the nucleosomes stacking on top of one another. And the DNA would be wrapping around the nucleosome. And that shape would be the 30 nanometer fiber. That's the diameter it would take. Now here's something else we need to know. What did I mean by nucleosome and the wheels and the doubling of proteins? What does that all mean? So essentially we have these two discs or two wheels that are back to back, and each is made up of 4 different histone proteins: H2A, H2B, H3, and H4. Now when I say each protein doubles itself, making a total of 8 proteins, we form an octamer. What do I mean by double, though? So let's imagine this is a histone wheel, and this is another one. We have 2 cookies here, our Oreo analogy. This wheel is made up of H2A, H2B, H3, and H4. This wheel is also made up of H2A, H2B, H3, and H4. What I mean is, if H2A is duplicated, its duplicate copy will be on this wheel. You will never catch the same protein doubled on the same wheel because they won't fit together. So we have one copy of H2A on this wheel, the other's over here. One copy of H2B on this wheel, the other's over here. One copy of H3, it's over here. One copy of H4, it's over there. That's how that's going to work. Excuse me. So who else is involved in this? So like we said, we have the 2 cookies that are our nucleosome. We have a histone wheel here and a histone wheel there, back to back. Now, where does DNA come into play? DNA is actually going to be this cream in the middle here. So the cream, not the best analogy, but I like Oreos more than the spool and thread. The cream is going to be the DNA in between these nucleosomes here. Now, like I said, a better analogy would probably be a spool and thread, where the spool is the nucleosome and the thread itself is DNA, where the DNA will continuously wrap around and around and around and around, and you have this tight formation. I like Oreos more, though, so we're going to go with Oreos. So how does this even work? What even happens? So we have a histone that's positively charged and DNA that's negatively charged. Having opposite charges is going to cause the bond to be strong, which is going to make this tightly wrapped thread in a spool complex. Now, we need to access DNA. Pretend we need to access DNA somewhere up in here. Somewhere even like in here or over there. Somewhere in here where we can't reach it. We could probably neutralize the positive charge, which will allow the DNA to be released. And the nucleosome will be able to be moved. We can push the nucleosome out of the way so we can get to the DNA. But how do we even do that? What is involved in this process? We need the CRC. The CRC is a complex. It stands for chromatin remodeler complex. That's going to use brute force to push the nucleosome to another portion of the DNA strand. Now, once this nucleosome is pushed, adjacent DNA nearby will be threaded onto this nucleosome. Because once you push the nucleosome from its original spot, the DNA from that nucleosome at that particular location will be removed. And once you push the nucleosome to a different portion of the DNA strand, that DNA in the new location will be wrapped around the nucleosome instead. So now by breaking ATP to release the energy, you can shift over the nucleosome. I like to think of this as a boulder and a bodybuilder. Our bodybuilder is our CRC, and the boulder is the nucleosome DNA complex. Once you neutralize the positive charge here, you can still, it's still a boulder. It's still something that can't be moved by itself. You need something to move it. So the chromatin remodeler complex, our bodybuilder, for example, is going to be able to take this boulder and just kind of whoop that way. One sweep across the DNA strand. New DNA will be adjacently threaded, and the DNA that was once here will now be accessed. Now, what about these histone tails? What even is a histone tail? We have to imagine it's like a sweater. The thread that's used to make this sweater is DNA. Now, imagine at the end of the sleeve, we have one little piece of thread hanging out here. That little thread, the extra thread, is going to be known as a histone tail. Now, these tails are rich in lysine, and lysine is a positively charged amino acid, and it's going to be an important player for the next step. So what is a HAT and what is an HDAC? HAT stands for histone acetyltransferases. These do exactly as their name tells them. They transfer acetyl groups to the histone. Easy peasy. So they're going to transfer acetyl groups to the nucleosome tail. That tail has a positive charge with the lysine. And once you transfer acetyl groups there, you're going to neutralize that positive charge. And that's how you make the DNA loose from the nucleosome. You're able to break that bond between the two. That's going to make your nucleosome free to move, and then you can make DNA accessible. Now, HDACs, on the other hand, do the exact opposite. They deacetylate the histone. So they're going to come in and remove the acetyl groups from the histone tails, which will restore the positive charge. And that's going to reinforce the bond between the DNA and the nucleosome. So now the nucleosome and the DNA are locked in place again, and you can no longer access DNA. I like this visual that I found. It's the kind of like pathway that we see. So you start with your nucleus. You go inside the nucleus to see all this loose chromatin, but it's packed up tightly in this nucleus. But if you give this chromatin a shape, condense it down into chromosomes, that's what we got here. Now you can look right at the end here, and we see, oh, look at that. We have our nucleosomes here. This is what keeps our DNA organized. We have all these little nucleosome balls, I guess, or wheels, or whatever you want to call them, that are keeping DNA interacted, keeping them very organized and very, like put away in a place you can find them again. Now, the difference between this and this is that this is heterochromatin. This is silenced DNA. You can't access this very easily because they're all so close together. And this is euchromatin, or active DNA. You can access DNA very easily through this step here because everything is all spread out. Now, I went ahead and drew, or tried to draw, the image of the nucleosome. We want to move this nucleosome in the direction of the purple arrow. We want to get this guy to go over here. The first thing that we need to do is call in a HAT. We need to call in HAT to come onto these lysine-rich nucleosome tails, which are positively charged. We need HAT to come in, plop some acetyl groups on all of these tails, which will remove or neutralize the positive charge, which will allow this to be free. CRC, or chromatin remodeler complex, can now come in and push this out of the way. We can get to where we need to get. Once that's all done and dandy, we can call in an HDAC. HDAC is going to come in and remove the acetyl groups from these lysine tails. That's going to restore the positive charge, and that's going to make DNA and the nucleosome locked tightly together again, which will make DNA inaccessible once again. Now we can jump into DNA replication, our second topic. So, this is where DNA genomes are faithfully copied, and by faithfully I mean we need to make as few mistakes as possible. This is a super duper complex process, but cells are stupid, and let's never forget that. That is the takeaway of this whole eReport. Cells are stupid, but everything that the cell does in all 8 of these topics is all regulatory. It's all signals that are given. So where do we begin? We need to start with the origin of replication. This is a specific sequence of DNA that is suitable for the beginning of replication. In eukaryotes, it's the Tau box. This is a region of DNA where the hydrogen bonds are very, very easy to break apart. This requires the least amount of energy to break apart. So we've had to find that sequence, Tau. Once it's found, DNA helicase is going to jump in there and start to unzip the double helix, separating our double strands into single strands. Now, something I forgot to throw into this slide, but I feel is crucial to say right now, is SSBP, or single-stranded binding proteins, are going to come in at the same time and hold these single-stranded DNA molecules apart. Because naturally, they're going to want to snap back together. But if they're together, we can't replicate DNA. So once helicase comes in and does its job, the SSBPs are going to hold the single strands apart, which will allow us, or allow DNA polymerase to come in and synthesize new DNA. But now we're talking about DNA helicase here. Once helicase comes in and unzips the DNA, we're going to form this bubble. It's called a replication bubble. Now, at either end of the bubble is what we call a replication fork. It's simply a Y-shaped junction, which is the active site of replication, and each fork is going to move in opposite directions. Opposite directions, moving away from each other, we refer to that as bidirectionality. 2 directions. Easy. Now, DNA is going to read from 5' to 3', normally. And double-stranded DNA has one strand that does that, 5' to 3' left to right. And the other strand is actually going to read 3' to 5' left to right. Another way of saying that is 5' to 3' right to left. It means the same thing, but in essence, it's all anti-parallel. DNA is anti-parallel. Now, this image is something I found on the internet. I thought it was very simple and very easy to understand. This top strand here, this top parent strand that has 2 bubbles in it, is running 5' to 3'. The bottom strand that's complementary to the top strand runs 5' to 3' in the opposite direction, which is good. That means they're anti-parallel. Now we see here helicase already came in and made our bubble. And at either end of our bubble, we have 2 junctions. Our Y-shaped junctions are replication forks. And this is where DNA replication is going to happen. It's the active site. Now these blue dots here in the middle of the bubbles that are kind of hooking onto those bases there are the new synthesized daughter strands by DNA polymerase. If you look down here, this is the end product. We have one parent strand in black up here. And we have another strand, but it's made of blue dots. That's our daughter strand that is complementary in base pairs and runs anti-parallel. The same thing goes for down here. This strand's running 3' to 5' this way, and it's a parent strand. And the daughter strand is running 5' to 3' left to right, complementary to this one. This is called semi-conservative. It's where you take one parent strand and you use it as a template to synthesize new DNA, or a daughter strand that's complementary. Now, something we need to remember, to make clear, I suppose, is let's think of this as a zipper. This is DNA helicase right here. And this is DNA itself. This is now a single-stranded DNA molecule, and this is also a single-stranded DNA molecule. In order to better visualize this, we have helicase that starts way up here, and these 2 strands are actually together. Once DNA helicase is dragged this way, all the way down, these double strands now become single strands. In essence, that's what happens when we initiate replication. We have DNA helicase on a double-stranded track that slowly unzips it to form this bubble. Now, how is DNA synthesized? Well, we already discussed that a little bit. DNA polymerase is going to come in and use semi-conservative replication, which is where you take one parent strand to generate a new daughter strand. So then where do these guys come in? What are DNTPs? DNTP stands for deoxynucleotide triphosphate. This has one hydroxyl group on the 3' carbon and 3 phosphates attached to it. So when the phosphate bonds are cleaved, when you take those 3 phosphates and you break them, you break the bonds, you're releasing ATP. And this energy is what DNA polymerase is going to use to catalyze the process. If we're reading DNA 5' to 3', it must be synthesized the same way. So I've gone ahead and tried to draw the replication fork, and I have the parent strands drawn out in black here. This bottom strand is running 5' to 3' left to right. Easy. That means the daughter strand needs to be synthesized 5' to 3' going the opposite direction. So the thing about polymerase, DNA polymerase cannot make DNA from scratch. It needs some kind of template or something to sort of kickboard off of. So we have these RNA primers. RNA polymerase can make DNA from scratch. And what it's going to do is it's going to put down this primer here. And this primer is going to be used by DNA polymerase to synthesize new DNA all the way across. All the way. This strand is known as the leading strand. We have one primer here, and DNA can be synthesized in one continuous shot. That is what the leading strand is. It's when you can synthesize DNA without any breaks, and you can do it in one shot using one primer. Yay, hooray, right? No, well, the top strand can't do that. The top strand is running 3' to 5' left to right. And it's going in this direction. If we're reading it correctly, we have to go right to left. In this case, because 5' to 3', we need to go this way, which means DNA must be synthesized that way. So as this fork opens up more and more, we're going to have a primer here. If the fork opened up here, we're going to have a primer here, and we're going to synthesize DNA right here. The fork opens up again right here. Put a primer here, new DNA here. It'll open up again. Put a primer here, or rather right here, new DNA here. So as this fork continues to open in this direction, we're going to have to place primers and then do new DNA, and then another primer and new DNA, and then another primer and new DNA, all the way down this strand. If we're synthesizing in this direction, it is going to be literally impossible for us to do it in one shot because of the directionality. That's why this strand is known as the lagging strand. We need fragments of DNA to be synthesized over and over with multiple primers. Now, these fragments are known as Okazaki fragments. They're named after the scientists who discovered them. So just to recap, we have our fork. This is the leading strand because it's continuously synthesized. This is the lagging strand because it's synthesized in fragments. Now we get to the clamp loader and the clamp protein. Woo, excuse me. So usually, DNA polymerase would synthesize a base one at a time, and then it'll float away. And it'll come back, put one more, and float away. Come back, put one more. That's going to take way too long. DNA polymerase doesn't understand that we don't have time to waste. So, my friend the clamp protein understands we have to, expedite this process. So what clamp protein does is it'll lock around DNA, loop around DNA like a ring. And that ring will interact with DNA polymerase, which will keep it from floating away. Brilliant, genius, I love it. But even more important is this guy here, the loader, the clamp loader. The clamp loader is what's going to open up the clamp protein, open up that ring, which will allow the ring to then lock around DNA, which will allow that ring to interact with DNA polymerase, which will keep it from floating away. It's genius, I tell you. I call the clamp protein the time saver protein. Why? Because DNA polymerase would literally be there forever if we didn't have that. The clamp protein is going to expedite the replication process, which I think is brilliant. Now, we have to tie it all together. We start off with DNA helicase unzipping the double-stranded DNA at the replication origin. All these proteins are going to be attracted to the bubble once the bubble is formed with DNA helicase. But even before that, we have to make sure that the SSBP, or single-stranded binding proteins, come in to hold those single strands apart, because without them, DNA would just snap back together. Now the clamp loader is going to open up the clamp protein. The clamp protein will loop around DNA like a ring. That is going to interact with DNA polymerase, which will keep it from floating away. DNA polymerase will then continuously synthesize DNA, and on both parent strands, it will have to use RNA primers, whether it's once or multiple times. And that will mean one parent strand is leading and the other is lagging because it has to be synthesized 5' to 3', depending on directionality. Now, once these primers are placed down, on both the leading and lagging strands, and the entire process is complete. DNA ligase has to come in, which is known as molecular glue, and it'll just make ends meet. Kind of funny, but also, yes, literally make ends meet. The key about DNA polymerase now is it can do this thing called proofreading. It's not just a synthesizing enzyme, it has the ability to check its work in real time. So as it's synthesizing bases 5' to 3', it's proofreading 3' to 5'. It's checking its work to make sure it's doing what it has to do, making sure it's putting the correct bases in. Let's say it caught its own mistake. If there's an offending nucleotide in the strand that it just synthesized, it's going to move that base to a separate domain of its enzyme, of its own self. It will cut that phosphodiester bond. It will remove the nucleotide that's wrong, and it will put the correct base in. And then the DNA strand will be on its merry way. How amazing is that? This provides a beautiful jumping point into topic number 3, which is DNA mismatch repair and double-stranded DNA breaks. But we're going to start off with the first half, DNA mismatch. I call this the safety net repair mechanism because it's used only when DNA polymerase misses its own mistakes, aka safety net. So similar to DNA polymerase, this repair mechanism, this mismatch repair system, is going to find errors in the backbone of DNA. It's going to feel for distortions. That's what polymerase does while it's synthesizing and proofreading. It will feel the backbone of DNA to make sure that everything is correct. If it feels a distortion, if it knows something is wrong, the DNA mismatch repair system will take out an entire section of the DNA that includes the mismatch base inside of it. That entire section will be removed. And then the old non-mutated strand will be used as a template to synthesize the correct nucleotide. So what I did here is I just kind of color-coded the bases here. Guanine is yellow, cytosine is blue, thymine is green, and adenine is red. I've already gone ahead and I've circled the offending nucleotide here. And because this is mismatch repair and not polymerase proofreading, the entire chunk of DNA right here, this entire section, is going to be taken out right over there. It's pulled apart over here. And this blue, we'll look across here, it's across from a green. It should be a red. It should be an adenine. So this section is taken out, and the correct nucleotide, adenine, is put in. Once we've fixed it, we're going to put it back in and ligate these ends together. And now we have the correct base. Easy peasy and super simple. So now we'll talk about dsDNA breaks and repair mechanisms. dsDNA break is when the DNA molecule is completely severed into two. This is lethal. We don't want apoptosis. We don't want to die. We just want to survive. So what are we supposed to do? What is the cell going to do? Well, luckily, the cell has two mechanisms of repair. One is non-homologous repair, and the other is homologous recombination. Now, the way I like to think about this is imagine you have a puzzle, maybe just two pieces of the puzzle. Let's say we have blue guy and green guy right here. If we want to do non-homologous repair, we can take this blue nub, file it down so that this edge is completely flat. And then we can take this green hole and file it all the way down so that this edge is flat. And then we have two flat ends we can just glue together. Easy peasy. Now, that's all fine and dandy, but you're losing genes that way. You could be losing something important. And this is almost like a last resort type of repair mechanism. And the only reason why it's last resort is because it's not the most efficient way to save yourself. That's why the cell prefers homologous recombination. This is the method that it'll try first. Homologous recombination is a little bit more complicated, and I'll tell you why. For this to work, we need a nearby DNA molecule which is extremely similar to the original strand. This is going to help us make a template. The similar strand that we find nearby will act as a template, and this will help us form a new strand to kind of fix that break. Now, I'll say it again because it's crucial. We need the similar DNA molecule for this to work. Because if we don't have it, the whole thing will fall flat. Now I've kind of went ahead and drew out a very simplistic version of homologous recombination. So these blue strands here, this one kind of loops down this way, but if it wasn't looped down, they'd be straight up here. It got broken, just slashed back like that. Bow, X-ray radiation or whatever we had happen here. We had a break. Now we have a slash in our blue molecules. The first thing we need to do is have nucleus come in and chew back this end of the 3' molecule, and also on this end too. Now these red strands down here are similar strands to the original. So what this piece here is going to do is it's going to start to survey and see if it can find similarities. This is known as a homology search. And once it does, it will begin strand invasion. This is it's close enough in proximity, and this strand is already looking for homology. So it's going to kind of like sniff around a little bit, and once it sees that this is nearby, it will just kind of come in here and invade this molecule. This strand needs to move out of the way. That's what's going to be known as the D loop. And while this strand moves out of the way, this strand here is going to act as template for this strand. This one sees it has homology with this one, and this one sees it has homology with this one. And it works out because they are running anti-parallel from each other. This is 3' to 5', this one's 5' to 3', and the same goes for the other two. So once they see that they can base pair, polymerase will come in, use this strand as template to sort of fill in this little gap here. The same thing will happen up here. Polymerase will come in and it'll use this strand as template to fill in this gap. Once that's all good and done, all these ends they'll get snipped, and this is our final end product, and we have homologous recombination. Our blue strand now has pieces of red in it, thanks to our similar strands that we had before. Now we jump into the assembly of the PIC. What even is PIC? It stands for pre-initiator complex. Now, it's in the name, pre-initiator. This is what we need to form before we can even have transcription occur. Now we can see here in this visual, we have our TATA box right here. We have the TBP, we have our transcription factors, we have an upstream enhancer, this is polymerase here, and this is our gene. Easy peasy. But what else needs to be there? Well, we need the promoter, and all those transcription factors that were denoted in purple are needed as well. Now, the promoter in eukaryotes is known as the TATA box, and that's because this sequence is thymine adenine, and the hydrogen bonds there are easier to break. This is very easy to break apart with the least amount of energy. Excuse me. So once the sequence is found, the TBP, or the TATA binding protein, I believe was in red, will latch onto the promoter. Once that latches onto the promoter, then all of our transcription factors will come in. And this is where DAB-Phe comes into play. This is just a clever mnemonic that we use to memorize the order. So general transcription factors, or GTFs, will play an essential role in the process, and these guys are what are going to help recruit RNA polymerase II to the promoter, and this will also help give the signal whether or not we want to initiate transcription or not. So the order that they arrive are in the name. DAB-Phe, TF2D, TF2A, TF2B, the dash or the space in the middle represents polymerase coming in, TF2F, TF2E, and TF2H. This is the visual blown up a little bit. This is our promoter right here, the TATA box. Once the sequence is found, we have TBP, which will come in and latch on. We'll have a transcription factor that will, or a bunch of transcription factors that will come in. I wish they were labeled, but they're not. But we need to have DAB come in first, and then polymerase will come in and latch to the promoter, and then we'll have FE and H. Now, this is an upstream enhancer. This is supposed to be a little bit farther out in the gene, but it's doing this thing called looping out, where DNA will sort of bend itself all the way here to kind of cast its vote in telling the PIC whether or not we should transcribe this gene or not. And that's going to come in later. So we'll get into topic 5, which is post-transcriptional mRNA processing. So what even is an mRNA? It stands for messenger RNA. And when you transcribe DNA into RNA, you're really making mRNA. That's really the end game. And this process of transcription occurs in the nucleus. Translation occurs in the cytoplasm. Now, in order for mRNA to get out, to be, help in translation, it has to leave the nucleus. But cells are stupid. So how do they really know when to let mRNA out? It's all about regulation, order, sort of system that we have going on. So once we have mRNAs that have been transcribed, we have these things called 5' caps and poly A tails. These are what are known as our seals of approval. These are what tell the nucleus that these mRNAs are authentic, genuine, true, and mature. These guys are ready to be sent out to the cytoplasm for translation. These guys are real. They're not phonies. They're real. They need to be released, and they are ready for translation to occur. These guys are almost like little flags to tell the nucleus, hey, we're ready to go. Even more importantly, though, is circularization. Without that shape, you're not really an mRNA that's ready to go. And on top of that, we had to splice because in mRNA sequences, there are some portions that are just not important that we don't need. And those unimportant regions are referred to as introns. So we have to sort of splice them out. And the way that the cell, quote unquote, knows how to do this, I hate using that word, but the way the cell knows is that every single intron contains a conserved sequence in the beginning and at the end. And these sequences serve as molecular cues for splicing. Because again, cells are stupid, they don't know. So I guess these conserved sequences are signals for the cell to splice the mRNA. Now that we have it spliced, now that it's circularized, has a 5' cap, poly A tail, it's ready to go into the cytoplasm. Now we hit translation, and literally we're going to talk about all of it. So the purpose of translation is to translate nucleotide language into amino acid language. And what that means is we're going from RNA to protein. And RNA is sort of like our bridge. And DNA and RNA nucleotides that are in groups of 3 are referred to as triplets or codons. Now, there are 64 possibilities, but we only need 20. We only have 20 amino acids in our codon alphabet, I suppose. But this abundance allows for redundancy and protection of the genetic code against mutations that can affect us, which is good. That's a good thing. Something important to remember is reading frames. In order to read the codons correctly, you need to take a certain frame. A certain perspective must be used. So there are 3 potential reading frames, but only one is correct. And the one that's correct is the one that starts with the sequence AUG. The importance of AUG is that it's the start codon. AUG will always translate for the amino acid methionine, always. That's why it's the start codon. You continue reading from that AUG point in groups of 3 until you hit either UGA, UAA, or UAG. And these are stop codons. Now, this visual is something that I found really, really fascinating. I didn't even realize that there were other characters here, but there's English so we can understand. We have an mRNA strand right here. And we want to read it correctly. We have 3 potential frames. We have a blue underline, we have red slashes, and we have green slants. Now, if we look at the blue, we see, okay, we start with AUG, AUA, CAU, etc. The red says UGA, UAC, AUA, CAU, UCA, etc. And the green says AUA, CAU, ACA, UUC, etc. Which one is correct, though? The correct one would be the first one, reading frame number 1. Because the crucial detail for reading frames is you need to start with AUG. This is crucial. This is our start codon. This is where we begin. You find the sequence first, and then you continue to triplet all of your codons together from that point on. Now, what is tRNA, though? tRNA stands for transfer RNA, but we prefer to call it translate RNA. Because the codons in mRNA don't directly bond to their corresponding amino acids. Not directly. The thing about tRNA is that it has a 3-base anticodon on one end, and this region will directly base pair with the codon on the mRNA strand, which will provide recognition of the genetic code. How genius is that? This is the bridging molecule. This is the only one that can speak both nucleotide and amino acid language. It can do both. So what exactly is going to happen here? We have ribosomes, and these guys are what are crucial to translation. Ribosomes are essentially RNA and protein molecules that will come together to form a complex, and it has one responsibility. Now, there are two subunits in ribosomes. There's the large and the small. The small will match the mRNA codons to their proper tRNA. So like I said, on the mRNA strand, which is denoted in the black line here, the mRNA will have these green codons. And tRNA has the anticodon that will base pair with the codon. The small subunit of ribosomes will sort of aid in matching these guys together. Now, the large subunit of the ribosome will form polypeptide bonds between the amino acids, linking them in a chain. So there's this organized system that's going on here, and let's take a closer look at that. We see A, P, and E. That stands for A site, P site, E site. These are voids within the larger subunit that will allow for the tRNAs, the amino acids, and the polypeptide chains to interact in a smooth and organized fashion. So before we can even begin, we need the initiator tRNA. What's so important about it, though? So the thing about the P site is that only tRNAs with a polypeptide chain attached to it can enter the P site. But the initiator tRNA is the only exception. It is the only tRNA that can enter the P site directly with only one amino acid, because we have to start somewhere. So the thing about the 5' cap that we discussed in the last unit, excuse me, is that this 5' cap on the mRNA will bind to the small subunit of the ribosome. Once it binds to that small ribosomal subunit, the tRNA will then search for the start codon. This is why the start codon is so essential. In order to get the correct reading frame, you need to find that AUG, that first codon that will kick off everything. So when TNA is looking for that start codon, the mRNA 5' cap will bind to the small subribosomal unit, and then this guy will go looking. Once we find it, then the large ribosomal subunit will join. So once the start codon is found, then and only then will the large ribosomal subunit join in. So essentially we have the smaller subunit and the larger subunit of the ribosome sandwiching the mRNA strand. So we have the initiator tRNA in the P site, and then we have the large ribosomal subunit come in. The small has already been there. Once we have these two sandwiching mRNA with the tRNA in the P site, we'll have another tRNA enter the A site with one amino acid. A polypeptide bond between the tRNA in the P site and the tRNA in the A site will form. And then every single tRNA from that point will shift over one site. So here we go with the full-on steps. So we have tRNA with the correct anticodon carrying the appropriate amino acid. It'll enter the A site and bind to the codon via base pairing. We already know that the tRNA carries an anticodon that matches the mRNA codon. Now, the amino acid on this tRNA, the tRNA that's in the A site, has an amino acid. That amino acid will form a peptide bond with the amino acid chain linked to the tRNA in the P site. Now, this will transfer the entire amino acid chain to the tRNA. The entire ribosome will shift forward by one codon. The tRNAs will stay base paired with their codon. So essentially, it's not the tRNAs kind of like lifting up and moving over. It's the ribosomal subunits that are shifting along the mRNA. So once the once the subunits of the ribosomes shift over, it will look like the tRNAs have moved over, but really the ribosome itself has moved, which is essential. So what's going to happen is the tRNA in the P site will end up in the E site, tRNA in the A site will move to the P site, and newer incoming tRNA will enter the A site. And like we said before, initiator is the only one that can fit into the P site with one amino acid attached. So now we get into termination. Stop codons, which is our UGA, UAA, and UAG, these are needed to end translation. But even more crucial are the release factors. These guys are what catalyze the release of the polypeptide chain from the tRNA in the P site, which will free it from the ribosome. So once we find this stop codon, the release factor will enter the A site, and this is our signal to the ribosome and to the tRNA to release from one another. And once the signal has been given, it means translation has been completed. Bada boom. No more, we're done. That's it. So this is a better representation, I think, of translation. I think this one is a little bit more sophisticated. It's also upside down, which I think is the correct way, but we've been doing it this way. I think I'll look at this one. So these red blocks here are our mRNA strand running 5' to 3'. We have our small subunit of ribosome, and we have our large subunit of ribosome. This is our start codon. This guy's going to latch on right in there, and notice it's in the middle-ish. That's our P site. This is the only tRNA that can fit in. It's the one that codes for methionine. Now we'll have another one, another tRNA with a new amino acid join into the A site. And now these two amino acids will form a bond onto this tRNA. This guy and this guy will connect, and these two will form a chain onto this tRNA. The entire ribosome will move over. Oh man, the thing is in the way now. But the entire ribosome will move over. So now the original tRNA that was in the P site, our initiator, will now enter the E site, and it'll float away. Now we have a tRNA with a chain in the P site, which is not breaking the rules. That's correct. That's what we need to have. And then a new tRNA with a single amino acid will join the A site. These guys will form a bond onto this tRNA. This guy will move over, float away, and then we'll have a polypeptide chain tRNA in the P site. A new one will come in the A site, and it's just a forever process until we hit our stop codon. Now we have topic number 7, combinatorial control and the mediator complex. So combinatorial control is essentially where we have groups of regulatory proteins working together to precisely control the expression of a gene. If we think about that a little bit more in depth, it's almost like a democratic vote, isn't it? So each regulatory protein will transmit its own vote. Look at that. Bada boom. It's like Congress. We got some kind of law that has to be passed. Should we pass it? Should we not? Raise your hands. Send the signal. Let me know. It's just tallying up votes. And then once we have a consensus, or like all the votes have been tallied up, only one signal is transmitted to the big honcho. So how does this relate to transcription? So when the PIC is formed, the pre-initiator complex is formed, we have all of our necessary GTFs that will recruit the RNA polymerase II to the correct location, position it correctly, separating the dsDNA of the gene, and then launching the enzyme down the gene to transcribe. But I've said it so many times, I'll say it again. Cells are stupid. How do they know when to initiate this process? Well, they don't know. It's all about combinatorial control. It's all about these signals that have been given off. And this is where mediator complex comes into play. Mediator complex is this huge protein that will basically sit on the end of RNA-P2. And what I mean by sit on the end of it, I really mean it's right next to transcription factor 2H. It's the last transcription factor to come in DAB-Phe. It's right near the tail of RNA polymerase II. Mediator complex is essentially like blanketed over the entire PIC as a whole. And this complex will act as a relay complex. It'll listen to the signals from activators, and it'll pass them on to RNA-P2. So each vote is tallied by mediator complex. This is the counter of the votes. Each vote is tallied and one mass signal is sent to the polymerase. But more specifically, the signal is sent to TF2H, as I said before, which is near the tail. So if we vote yes, let's transcribe this gene. Mediator will tell H to phosphorylate the tail of RNAP2, and the enzyme will just go chugging along the gene. Easy peasy. But if the vote is no, the enzyme will stay put. Now we hit our last topic, and this is my favorite one. You'll see why. This is RNAi. This is a mechanism used by cells for gene silencing. Gene silencing? All right, well, what does that require? Well, we need double-stranded RNA. That's a little unusual, but it's efficient because when dsRNA or double-stranded RNA is recognized by RNAi machinery, all dsRNA and ssRNA with the same sequence is destroyed. Our cells have a hitman system, and I think that's awesome. Like, that's so cool. And what it was thought to be was that RNAi was probably an evolutionary thing that was used to destroy viruses because viruses are RNA. But seriously, we have a hitman system? Our cells have a hitman system? Like, search out for this sequence and go destroy it and it'll just do it? That's insane. So what do we really mean by this? What we really mean is if a double-stranded RNA molecule with your gene sequence is found by RNAi, you will never make a protein from that gene again. Never. Any mRNA that is transcribed from that gene will be immediately marked for destruction. Immediately marked for destruction. Target sighted, immediately marked for destruction. If you're an mRNA with a similar or the same sequence as that double-stranded RNA that's being hunted down, you have a red flag on you that says, "Hey, I match, but I don't want to match, but you're gonna get destroyed." That's insane. So what are the steps that go into this? So firstly, we have double-stranded RNA that base pairs with itself, roughly 21 to 23 base pairs long, roughly. If the dsRNA is too long, dicer, which is an enzyme, will cut it to the appropriate length of 21 to 23 base pairs long. Now, these fragments made by dicer are called siRNA, which stands for small interfering RNA. Now, the siRNA will bind to RISC, and RISC stands for RNA-induced silencing complex. And RISC will essentially take the scent, quote unquote, of the siRNA. And the double-stranded RNA is separated. RISC will use one of those strands, get that one scent from one strand, to find other strands that will base pair with it. So that when complementary base pair strands are found to the one that RISC has, it will be destroyed on site, which I think is absolutely fabulous. I've used two different representations here. We've got our double-stranded DNA, or RNA up here rather, and now we have dicer, our enzyme, which will cut it to size, and this fragment here is known as siRNA. One strand will be used by RISC. The other strand, we'll talk about what happens with that later. So once we have one strand taken by RISC, RISC will get the scent of that single-stranded RNA. And by scent, I mean what will base pair with this. And once it's base paired, or once it finds sequences that will base pair, it'll be destroyed. Those sequences that will base pair with the single-stranded RNA that RISC has will be destroyed. This sequence matches, you're dead. That sequence matches, you're dead. Another one matches, it's dead. No question about it, which I think is fabulous. The same thing is being illustrated here, a little bit bigger and I think on a more simpler scale. We have dsRNA up here. This is the enzyme dicer, which makes our fragments. These are siRNA. Now we have RISC, which will take one strand from these fragments, one strand, use it as a scent. Let's see what will base pair with this. It'll go chugging along mRNA, and once it finds base pairs in the mRNA, they'll be destroyed. How cool is that? So how exactly is this efficient? I don't understand, like, how can this be this quick? I don't understand this. Put some numbers in perspective. About 50 small interfering RNAs can be used to silence thousands of pre existing mRNAs. How is this possible? Amplification. So when Dicer cuts new fragments of small interfering RNA, one strand is used by the RISC, and the other is used by RDRP. RDRP stands for RNA dependent RNA polymerase. You can maybe guess what that does. RNA polymerase will synthesize new strands that are dependent on RNA. This is how we make more dsRNA. And therefore, you can repeat the cycle over and over, and more mRNAs can be destroyed. How fascinating is that? Alas, we've come to our summary conclusion section of our eReport. So just to top everything off, we covered chromatin compaction, the 30 nanometer fiber, what that is. We talked about the chromatin remodeler complex, HATs, and HDACs. Then we talked about DNA replication. We talked about the origin, the bubble, the fork, every enzyme that we need, leading versus lagging, and we even discussed proofreading, which gave us a good segue for topic number 3, which is DNA mismatch repair. We talked about dsDNA breaks and repairs, strand invasion, the homology search, the D loop. Then we hopped into pick, every single GTF that's involved, the order in which they arrive, RNA polymerase 2, and the entire purpose of the pick. Then we got into post-transcriptional mRNA processing, which makes mRNA genuine. This is where we get the 5' cap and the poly A tail. Then we discussed every single step of translation, from the tRNAs to the reading frames, codons and anticodons, A site, E site, P site, subunits of the ribosome, release factors, everything, the whole shebang. Then we discussed the definition of combinatorial control. We discussed what happens. We discussed mediator's role, how that works, where it is. And then last but not least, we discussed RNAi, we discussed Dicer, we discussed siRNA, RISC, RDRP, and we even touched up on amplification to explain how this is so efficient. Hopefully this was super clear, and hopefully you enjoyed it.